In a previous discussion, we looked at all the branches and subdivisions of the nervous system. We said that sensory information about changes in the environment pass through the afferent side of the peripheral nervous system in towards the central nervous system, where it gets integrated and decisions are made for how to respond to those changes in the environment. Those decisions then pass out the efferent side of the peripheral nervous system. If it goes out to our skeletal muscles to control voluntary movements and interact with the outside environment, it passes through the somatic side of the efferent peripheral nervous system. But if those decisions pass through the autonomic side, it's going to control our internal environments in an involuntary kind of way. The autonomic nervous system largely helps us maintain our internal environment. It does so by innervating cardiac and smooth muscles as well as glands, and because of that, the autonomic nervous system can speed up or slow down our heart rate, shunt blood from one area of the body to another, increase or decrease blood pressure, and control the release of hormones. Most of these actions occur without our awareness or without our conscious control, and because of that, the autonomic nervous system controls those things that happen automatically inside of the body. We're going to take a look at some contrast between the somatic side of the efferent peripheral nervous system and the autonomic side to help us get a better understanding of how the autonomic side works. Somatic neurons control voluntary movements by innervating skeletal muscles. They are single neuron circuits with the soma or cell body of that neuron in the central nervous system, and the axon can be very long, go out and synapse with skeletal muscles that are far away from the central nervous system. The primary neurotransmitter used by the somatic neurons is acetylcholine. Autonomic innervation is a little bit different because the autonomic nervous system uses two neuron chains to communicate with effector organs, whether it's cardiac muscle, smooth muscle, or a gland. The first neuron in this chain, called the preganglionic neuron, has a soma in the central nervous system. Its axon synapses with a second neuron at a ganglion. The second neuron in the chain, referred to as the postganglionic neuron, has its soma in the autonomic ganglion. Its axon then extends out and synapses with some kind of effector organ. That postganglionic neuron may use acetylcholine or norepinephrine as its neurotransmitter. Because of that, the autonomic nervous system can stimulate or inhibit the effector organs it's communicating with, whereas the somatic nervous system is almost always excitatory. The autonomic nervous system can stimulate or inhibit its effectors. Let's take a look at these different kinds of neurons. A somatic neuron is relatively large. Its cell body is found in the central nervous system. Its axon extends all the way out towards a skeletal muscle, and it uses acetylcholine as its neurotransmitter. Sympathetic neurons of the autonomic nervous system, on the other hand, are two-chain kinds of neurons, with the preganglionic neuron having a cell body in the central nervous system, and its axon extending out to some sort of ganglion, where it communicates with the second neuron in the chain. This postganglionic neuron can either release norepinephrine, or the preganglionic neuron may communicate with glandular tissue that releases norepinephrine and epinephrine into the bloodstream. Parasympathetic neurons, on the other hand, are also two neuron chains, with the preganglionic neuron having a cell body in the central nervous system, and its axon synapsing with the second neuron at some kind of ganglion. The postganglionic neuron here releases acetylcholine as its major neurotransmitter. The autonomic nervous system is subdivided into the parasympathetic and sympathetic nervous systems. Most internal organs are innervated by both, and the signals from both act antagonistically or in opposite manners. The two subdivisions of the autonomic nervous system are not only functionally different, they are structurally different as well. If we're looking at the parasympathetic side of the autonomic nervous system, the neurons here exit in a craniosacral kind of way, meaning the preganglionic neuron exit either near the cranium or at the end of the spinal cord down where the sacrum is. Because of the way it exits, the preganglionic fibers of the parasympathetic side are very long, whereas the postganglionic fibers are relatively short. And the ganglion are going to be located near the effector organs. The sympathetic side of the autonomic nervous system is structured in an almost opposite kind of way. Here, the exit sites are called thoracolumbar because they're exiting in the thoracic and lumbar regions of the vertebral column. Because of where they exit, the preganglionic fibers of the sympathetic nervous system are short, and the postganglionic fibers are much longer. Here, the ganglia are located near the spinal cord. This difference in their structural complexity also helps us with their functions being opposites of each other, because all of the preganglionic fibers use acetylcholine as their neurotransmitter. If their ganglia were located near each other, there could be miscommunication. A sympathetic signal could wind up communicating with a parasympathetic postganglionic fiber because all preganglionic fibers use acetylcholine as their neurotransmitter. Because the ganglia are found in different portions of the body, we don't get that cross communication. On the other end, with our postganglionic fiber, we also need to avoid cross communication. Because of that, sympathetic postganglionic fibers use norepinephrine as their primary neurotransmitter, and parasympathetic postganglionic fibers use acetylcholine as their neurotransmitter, so that when they talk to the same organ, they use a different neurotransmitter so that they don't have cross or miscommunication. And because those organs are receiving two different kinds of neurotransmitters, it can be a stimulatory or an inhibitory kind of signal that they are receiving.